So this is an example now of another way of assembly besides this sequential assembly. This is called concerted assembly, where things more or less tend to happen all at once, although there are obviously some precursors that have to be made. This is the formation of influenza virus. And we may have looked at a slide like this before, but now we'll look at it in some detail. And remember, the influenza virions, the RNAs replicate in the nucleus. There they get wrapped up in the viral proteins that are eventually going to end up in the virion particles. So we have genome replication, we have mRNA synthesis. The mRNAs have to go out in the cytoplasm, get translated, and then the proteins that are bound to associate with the viral RNA get shipped back into the nucleus by their import signals, of course. So now we have the formation here of a ribonucleoprotein. It's an RNA coated with 4 different kinds of viral proteins. That gets shipped out of the nucleus. It has an export signal, so it's recognized for export. It makes its way to the plasma membrane. Now, meanwhile, in this infected cell, the viral glycoproteins are being made by the secretory system. These are made on rough ER. These are ribosomes attached to the ER, translating the viral mRNAs that encode HA, NA, and the M2 ion channel. And these get shipped up to the plasma membrane, where they form these little patches. Right, patches of HANA and M2. And if you look at an infected cell early, you can see these patches. So the whole membrane is not covered with these viral glycoproteins. It happens in patches. You concentrate the material for faster assembly. So here you have a patch of HANA and M2. And the ribonucleoprotein, we'll find it. There are signals for the RNP to associate with the membrane and also with the viral proteins. And at some point, this whole assembly buds out and forms a virion. So that's why we call this concerted assembly, because the virion is formed all at once. I mean, the protein, some of the components are formed in different steps, but they all come together to form a virion. So that's called the budding process. And of course, you can see how in this budding process, the virion gets its membrane, its lipid envelope from the host cell, and you can see how the viral glycoproteins get incorporated into it. So that's how an envelope virion is made, at least at the plasma membrane, and that's influenza virus. The driver, one of the main drivers of budding is the glycoprotein, which I said if you express on its own will drive particle formation. So this is a schematic of the influenza hemagglutinin. Remember, it's one of the 3 proteins in the virion, hemagglutinin neuraminidase and the M2 ion channel there. So here's the HA shown in a linear form. It's a glycoprotein. It is membrane bound, as you can see. It has a lot of sugars attached to it. These are the sugars, these Ys. It has disulfide bonds. Of course, it has a signal sequence because it's translated into the ER. There's the fusion peptide, which you remember remains near the membrane until it's ready to fuse. It is released by cleavage to make the N-terminus free right there, and it's kept by the membrane and then pops up when the pH drops. So this is one of the viral glycoproteins that is brought through the secretory pathway to the cell membrane, the plasma membrane. As viral glycoproteins pass through the secretory pathway, they are subject to many of the modifications that any other protein would be subject to. So here's just a schematic of the typical secretory pathway movement from the endoplasmic reticulum all the way up to the plasma membrane. We're actually looking at the production and movement of the HA of influenza virus. So again, it's translated on rough ER. It has a signal sequence which gets it into the ER in the first place, and that's of course cleaved off to free the N-terminus of the HA. And then the molecule moves through the pathway, through the Golgi network, all the way to the plasma membrane. And as it moves through, various modifications happen. The protein oligomerizes, sugars get added and taken off, and finally the HA gets cleaved in some cases so that it is produced as a cleaved polypeptide as shown here, and will be active then in new infections. So here's the mature HA. It's a transmembrane with the cleaved HA here, and the fusion peptide buried down by the membrane. And curiously, the part of the viral protein on the inside of the virion has a bit of lipid bound to it. That's the yellow stick there, and that makes it stick in the membrane. And why that is not really understood, but apparently it's important. So another example of concerted assembly, formation of retrovirus particles. There's a few differences here I want to point out to you. These also form by budding, but they mature after release. Remember, influenza particle was mature as it was budding out, but these mature afterwards. So that's what I want to show you. Now remember, retroviruses, their mRNAs are produced from transcription of the provirus. The provirus, of course, being the integrated DNA copy in the nucleus. The mRNAs are produced, they get shipped out into the cytoplasm, and some of them get translated, of course, into various proteins. Here, for example, is the structural precursor. It's made as a polyprotein. And remember, to get the enzyme reverse transcriptase and integrase, you need to have some kind of anti-termination event, either frame shifting or suppression, to get a little bit of the RT made. So mostly you get structural proteins, and those will associate with the RNA genome. They will go to the plasma membrane where the viral glycoproteins have been inserted, just like they would be for influenza virus. You can see here the transmembrane glycoprotein is in the plasma membrane. The matrix protein, which will form that shell just under the membrane, assembles there. You can see it's put up as a very nice precursor. Matrix capsid will form the actual shell of the virion, and then nucleocapsid protein, the white one here, is what actually binds the RNA. So you can see the nucleocapsid binds the RNA genome, and this whole assembly, matrix, capsid, nucleocapsid, then goes up to the plasma membrane where it starts to form a shell. Then you have a bunch of these lining up here, okay, RNA, proteins, and then you have a little bit of production of the polymerase precursor, which gets also incorporated. So the reverse transcriptase is the blue one here. The integrase is purple. So you can see a few of those get incorporated into the virion as well, because they're part of this polyprotein. See the matrix, capsid, NC, protease, RT, integrase, that can go up there as well. This whole assembly buds, but it's not finished yet. It's not a mature virion. This, you can see, looks different from the final virion. Here there are some proteases incorporated into the particle. Those are the yellow molecules. And after the virion is formed, those proteases act and mature the capsid. So now you have that icosahedral-based capsid in the interior forming. Okay, so that's an example of maturing after release. Otherwise, it's a similar process of influenza virus, a concerted form of assembly by budding. And the glycoprotein of the retrovirus is illustrated here. It's very similar to that of the HA. It's a transmembrane glycoprotein, so it's produced in the ER. It moves through the Golgi. It gets glycosylated. As you can see here, the signal peptide, of course, is released. There's an internal fusion protein, which is liberated by cleavage at these orange circles, and it folds properly in the ER Golgi to look like this. So on the surface of the virion, it looks like this. Here's the fusion peptide bent close to the membrane, so it won't randomly fuse with any membrane. The SU is the attachment protein, and we talked about how this works when it binds receptors and leads to fusion at the plasma membrane. Now, some of these proteins get modified with lipids as another way of telling them where to go in the cell. So we've talked about signal sequences that bring proteins through the secretory pathway, but there can be lipid modifications occurring as well, and these typically target proteins to membranes of different sorts. Here I'm showing you three different kinds of lipid, myristate and farnesol being attached to a protein, and also geranol, geranol, and palmitate. These are just different lipids that can be covalently linked to viral proteins. Okay, and they get modified typically to point them to membranes. So it's another way of addressing where a viral protein is going to go by putting lipid onto it. So here, for example, is what lipid modification does during retrovirus assembly. This is the pathway we just talked about. And I was telling you how this capsid precursor gets to the plasma membrane and brings all the proteins and RNA with it. Well, it doesn't just magically go there. The matrix protein at its N-terminus is modified with a lipid. It happens to be myristate, but the point is this lipid is covalently linked. It makes the N-terminus hydrophobic, and that's what directs the precursor with the RNA attached to it to the plasma membrane. If you mutate the amino acid to which myristate is attached, so that myristate can no longer be attached, you will not get production of virus particles. So this myristate is essential for particle formation. And again, because it helps target the matrix protein, the first part of this polyprotein, to the plasma membrane. Now, also part of this precursor is the capsid, which forms, of course, the icosahedral capsid beneath the M protein. And you can see nucleocapsid. Nucleocapsid is the protein that binds RNA. It's what latches onto the viral RNA and brings it up to the plasma membrane along with the structural proteins. So you can see it's a very orderly process. And that viral RNA, that nucleocapsid has very specific RNA binding sequences within it. You can see them right here that allow viral RNA to bind to it and nothing else. You don't want to bring cellular mRNAs into these virions. That would be a waste. So there's a specific signal for incorporating these viral RNAs.